And last but not least, we're going to have Tim Lowinger, who's been Chief Scientific Officer at Mersana since 2008, where he has led all of Mersana's discovery efforts focused on innovative approaches to ADCs. Prior to joining Mersana, Tim acquired nearly 15 years of experience in drug discovery while at Bayer Pharmaceuticals, including in the United States, Japan, and Germany, where he contributed as co-inventor of multiple drugs. Tim earned his BSc and PhD degrees in synthetic organic chemistry, at the University of British Columbia, and was a Merck postdoctoral fellow with Leo Paquette at Ohio State. Let's welcome Tim.
Well, it's sincerely a pleasure to be here and speak to you about some of the work that we've been doing at Mersana, and I'd like to thank the organizers for the honor to participate here. So I'm here to talk to you about a STING-agonist ADC platform, and what you see here on the screen is not a STING-agonist ADC. This is not a bait and switch, but I can't present in a ADC symposium that includes both Carolyn Bertozzi and Peter Center without telling you just a very brief amount about another drug that we have in the clinic at Mersana called amiltatug ledadotin, or Emily as we call it. But as you see in those circles, we have incorporated bioorthogonal click chemistry as part of this molecule. We have our own novel auristatin, but both of these, of course, were inspired by the work of Carolyn, Peter, et cetera. And so what we're hoping to do at Mersana is, stand on the shoulders of giants and hopefully try to innovate and make a difference for patients as well. Very quickly, we did just recently disclose some clinical data on Emily at ASCO. It is a B7H4 targeted ADC, and B7H4 happens to be highly expressed in a rare tumor type called adenoid cystic carcinoma type 1. Now, this is a tumor type that unfortunately is very aggressive, and there are no approved therapies for this. It often occurs in young adults, and the progression-free survival with traditional chemotherapy is on the order of weeks. We were very pleased to share this data and to obtain this data clinically, where you can see in 9 different ACC1 patients that are not pre-selected for B7H4 expression, but again, are likely to be B7H4 high because of the biology of this disease. 8 out of 9 showed tumor shrinkage. 5 out of those 9 achieved PRs so far. And we see progression-free survival on the order, not yet reached, but approaching a year. And this is just one of those patients. This is a 27-year-old woman, newly diagnosed with ACC. She had a 2-inch lesion in her lung where it had metastasized. And you can see on the right-hand side here, after 5 cycles of Emily, that tumor had completely disappeared. So we're excited about the potential for this drug. The clinical trials are ongoing. And I should also maybe mention that together with this efficacy, we're seeing a profile where we have no concerning neutropenia, peripheral neuropathy, or ocular toxicity. But now for the main topic that I'm here to speak about, which is STING-agonist ADCs. So I think everyone here would agree that immuno-oncology has changed the way that we treat a number of cancers. And we've seen real breakthroughs with the checkpoint inhibitors, et cetera, that really focus on adaptive immunity. But what we wanted to do with an ADC approach is see if there was a way to incorporate the innate immune response as an anti-cancer approach. We deliberately decided to focus on STING because we believe STING is a fundamental immune pathway, and there's a wealth of data, clinically as well as preclinically, to support the importance of the STING pathway. Now, we felt that an ADC approach was an ideal approach for targeted innate immune activation with STING, because when you think about it, what you're trying to do here is unleash a very potent mechanism in a local fashion. And that's exactly what we're trying to do with ADCs. Others have tried to take advantage of STING through, for example, intratumoral injection, where you can hopefully get that localized effect. But of course, not all tumor lesions are injectable. Some other companies have taken a different approach where they hope to find the therapeutic index by still dosing systemically, but there you risk that you'll get systemic activation and toxicity before you can achieve the anti-tumor efficacy. But with an ADC approach, we felt that, again, something which everyone here is very familiar with, you can have that magic bullet effect where you can deliver directly through systemic administration to all the tumor lesions while sparing healthy tissue. So that was our goal. It's very important on how and where you deliver STING to maximize the therapeutic index. And again, that's a major advantage of an ADC approach. With free STING agonists, for example, if you're injecting it directly into the tumor, you're relying on diffusion across the cell membranes into the cells in that tissue that you've injected. However, that's passive and indiscriminate diffusion, and unfortunately, a cell membrane, whether it's on a T cell or on a tumor cell, probably looks the same to that payload. And we know from the work of others that certain cell types respond beneficially to STING activation, but others, such as T cells, are actually killed by STING activation. With an ADC approach, our hope was that we could leverage the precise delivery through an antibody-directed delivery to deliver to antigen-expressing cells through antigen binding and internalization and STING payload release, but secondarily, through avidity-enhanced Fc gamma receptor-mediated uptake, we could deliver into the tumor-resident immune cells and activate in those cell types as well. And then because T cells don't express the antigen, they don't express FcR gamma, we, in theory, would be able to avoid delivery to those cells as long as we designed our ADC appropriately. So to set out on this, it was actually a very big endeavor because first we had to say, okay, what about a STING agonist payload? Where do we start? And again, here we were inspired by the work of others. GSK had disclosed a small molecule STING agonist that they were delivering systemically, and that was the inspiration for our starting point. But we wanted a STING agonist payload specifically for an ADC application, which would be different than for a systemic application. And so there were certain parameters that were really crucial for us. We wanted a highly potent STING agonist. We wanted it to be active against all the major haplotypes. We wanted it to be active versus mouse, rat, NHP, and human to facilitate translational biology. But importantly, with an ADC approach, we wanted very low cell permeability. We wanted to control where we put that STING payload via our antibody. And once it's released there, keep it there. And then once that cell was killed and it's released systemically, we wanted to have a very short half-life so it could be cleared from the system and hopefully not cause systemic cytokine activation. And then finally, as I think we can all appreciate, physical chemical properties were important. You wanted to have a payload that was amenable to conjugation to an antibody. So we identified that payload, and you'd think, well, okay, you're 99% of the way there. No, that was far from the case. Once you have a new payload, what we felt was very important, based on our previous experience, was to develop what we call the scaffold and linker precisely designed for the physical chemical attributes of that payload. Our proprietary STING agonist is still a relatively greasy molecule. So we looked at a variety of different parameters, including hydrophilicity, enhancing moieties, such as the PEG-bisglucamine that you see here, different linkers. We looked at cleavable linkers, non-cleavable linkers. We looked at different methods to bioconjugate, and we found that those parameters were very important. You can see different examples shown there until we ultimately determined that this was the best molecule to take forward. You'll see there that it has a maleimide. We're doing cysteine, old school bioconjugation. And you might wonder why. You just showed me that you're using orthogonal chemistry in your other molecule. This was actually very deliberate as well, because with the click chemistry that we had used in Emily, at asparagine 297, where you conjugate, if you conjugate this molecule with that technology, you actually block Fc gamma receptor binding and internalization. As I said earlier, and I hope to show in some of the subsequent slides, we felt that that was a very important attribute. The cysteine bioconjugation did not block Fcr gamma binding. So this is the first in vivo study that we did. And to evaluate this platform, we did, like many others do, we went to HER2 models. Again, we all know trastuzumab is a very well understood molecule. There's a lot of models available. And so we went into this HER2 high SCOV3 model. And you can see a number of comparators. We have our targeted ADC in blue. We've got a control ADC in gray, exactly the same linker payload scaffold, just conjugated to an antibody that does not bind HER2. We've got the unconjugated antibody, and we've got a free IV STING-agonist as a comparator. As you can see, all of the comparators are essentially inactive in the model, whereas a single dose of our trastuzumab STING ADC was sufficient to eliminate the tumors and give us durable regressions. Okay, so that's the first sign of efficacy, which we were very pleased about. But again, a key attribute was, could we improve the tolerability? What about systemic cytokines? So we looked at that in the animals as well. And here we see, again, the story that we were hoping to see, but very different than what you saw in the efficacy. As you'll recall, the free IV STING agonist in that previous slide wasn't efficacious, but here you can see that it is causing quite significant systemic cytokine elevations. In contrast, our targeted ADC, STING ADC in blue, shows low and transient elevation of STING. Again, exactly what we would hope to see by the benefit of an ADC approach. Okay, so that's the systemic cytokine profile. But what is happening in the tumor itself? In this case, we did a different experiment where we implanted tumors, human tumors, into immunocompromised mice. And at different time points, we excised those tumors, and we could measure both human cytokines and murine cytokines. And the beauty of this experiment is the only human cells in this tumor are, of course, from that tumor that's implanted. So that's the only source of human cytokine elevation. And murine cytokines must be from murine cells that are resident in that tumor sample that we've excised from the animal. And so what you see here with our targeted ADC, in contrast to the non-targeted control ADC, is a significant elevation of a number of different cytokines downstream of STING with the targeted ADC. And you see elevation of both human cytokines as well as mouse cytokines, suggesting that we are delivering to both the human tumor cells as well as to resident mouse cells within that tumor. So we wanted to understand this better. And again, we designed the molecule so that there wouldn't be a bystander effect. We believed that we could control an uptake into tumor-resident immune cells through Fcr gamma binding, but was that the case? So Nanya Settenba designed this experiment where what we did was we deliberately blocked Fc gamma receptor binding in the green line that you see here. We have the control ADC, an Fc mutant that doesn't bind Fcr gamma, but it still binds HER2, and then the fully intact targeted ADC in blue. And what you can see is that Fcr gamma mutated agent is still active, but not as active as the fully competent targeted ADC. And that's because we've blocked Fcr gamma binding, suggesting that Fcr gamma binding and resultant delivery into immune cells is playing a role in the overall efficacy. To understand this further, we did the in vitro experiments on the right. We're looking at tumor cell kill in a co-culture of PBMCs and the STING-agonist ADC. And you can see that in the blue lines, with the fully competent ADC, at 5 nanomolar, we have significant cell killing, whereas the Fc incompetent ADC in green doesn't show much activity at 5 nanomolar, but you have to go to 25 nanomolar to see similar levels of cell killing, suggesting that you have to push the tumor cell part because you're not getting the benefit of the immune cell part. And to further understand that this truly was through the effect of the tumor cells, Nanya did the experiment where she knocked out STING in the tumor cells. You can see that the blue agent is still highly active because it can still bind to the tumor cells. It's unproductive when it's internalized into those cells, but it has that Fc-enhanced uptake into the immune cells, so it's still active. Whereas now the green, unproductive when it gets internalized in the tumor, not able to get internalized through Fc gamma into the immune cell, is completely inactive, consistent with this dual delivery, targeted antigen to the tumor cell, FcR gamma mediated uptake into the mouse resident immune cells. We did look at this platform across multiple different targets. It's not only HER2 that it works. Here you'll see 6 different targets, 6 different models, all with the same platform. You'll see the control ADCs there, and we see similar results across multiple different targets, including targets such as those on the right that don't internalize very well. Another exciting part about immune stimulating ADCs is the potential for immune memory. So we wanted to evaluate this as well. That's what this model did. We, again, implanted mice with these are syngeneic mice. We implanted them with a HER2-expressing tumor. 6 out of 9 of those animals were tumor-free. We took those tumor-free survivors, and now we subjected them to re-inoculation with 2 different tumors. The one in blue that it had originally been cured of, as well as a naive tumor that the animal hadn't seen before. And we were very pleased to see that the blue tumor would not regrow, suggesting immune memory, whereas the naive tumor in red grew similar to vehicle control. So now we needed to choose what we were going to advance to the clinic. And we looked at a number of different targets, as I showed you some of the in vivo data, but we did decide to focus on HER2. It's a crowded space, but it's a well-studied space, a well-understood space, and there were a number of reasons that we felt that we might be able to make a difference there. So this is our first Immunosynthen development candidate. It's currently in dose escalation studies. In vitro, we see 1,000-fold improvement versus the free payload in terms of the ADC for cytokine activation. In vivo, we see robust activity at doses 1 mg per kg or less. And we also saw excellent PK in non-human primates for both the antibody and the conjugated drug. We then looked to evaluate this in a number of different models, HER2 high, HER2 low models, as you can see here. And we benchmarked it versus other agents, including a TLR7 immune activating ADC from Bolt Therapeutics. And what we were pleased to see is that it worked in HER2 high, it worked in HER2 low, and it also appeared that the efficacy was not necessarily Cmax driven, where splitting the dose at 0.3 mg per kg could give us really robust efficacy as well. Now, what we also did with our HER2 approach was, and we like to believe, at Mersana, that every aspect of an ADC needs to be thought about and carefully selected, right? It's the linker, it's the payload, it's the antibody, it's the site of bioconjugation, and another aspect is, with that antibody, we could have used trastuzumab. I showed you data with trastuzumab, but we deliberately chose to identify a novel HER2 antibody. We did this in collaboration with Adimab, and we found an antibody that does not compete with pertuzumab or trastuzumab for binding, which would allow for the potential for combinations of this agent. Because if you're on trastuzumab, you're not going to combine with another trastuzumab-based therapy. And so here on the right, again, SCOV3 model, But you see a combination where we have the ADC alone here in blue. We have trastuzumab that's essentially inactive in this model. But when we combine these two, you can see that benefit of that combination, giving us 9 out of 10 mice. We don't believe this would have been possible if we had used trastuzumab as our antibody. We looked at this in other models, HER2 medium model, a HER2 low model. We did it in combination with trastuzumab. We did it in combination with pertuzumab. In both cases, we see the benefits of those combinations. And we believe that those benefits are due to additional cross-linking internalization that is facilitating the drug delivery into those cells. Now, we also know that trastuzumab and pertuzumab, very important drugs, widely used drugs, but we know that the HER2 treatment landscape is changing, and we all know that Enhertu is a fabulous drug, right? That's really making an impact and has really sparked the excitement in ADCs now. So what about combinations with Enhertu? Now, despite how good Enhertu is, it doesn't work in every tumor, and it doesn't work for every patient. And this is a model that Daiichi had published in, the GIMT-1 model, that is relatively insensitive to Enhertu. And here you can see doses at 3 mg per kg, here, 10 mg per kg here. As Greg Thurber has taught us, probably the more relevant data is here. This is more clinically relevant, but it's modestly active in this. We were very pleased to see that in combination with a suboptimal dose of 2056, we could drive complete responses in combination with Enhertu as well. And then, in addition to combination potential with HER2-directed agents, when you're looking at STING activation, it's logical to think, if we're stepping on the gas of the immune system with a STING-agonist ADC, would there be benefit on releasing the brakes with an anti-PD-1 as well? We did those models, again, syngeneic model, and here what you can see is, again, the benefit of the combination driving greater efficacy than either single agent alone. In terms of tolerability, we evaluated this in preclinical studies in depth in non-human primates. Of course, our antibody was cross-reactive with non-human primates. Our payload is very active in non-human primates cells as well. And we were very pleased that at repeat dose studies at 36 mg per kg, multiple doses IV, it was actually determined to be a no adverse effect level. So in summary, our preclinical data that I've shared with you suggests that we have achieved at least our design goals as far as we could determine preclinically. We are now continuing in dose escalation, and I look forward, hopefully before too long, to be able to share some of our clinical results. And then finally, I just want to acknowledge, first and foremost, the patients that have been in our studies and their caregivers. That's who we're doing it for, and we really appreciate their sacrifices in enrolling in our studies. And finally, the multidisciplinary team at Mersana, across all functions. We wouldn't have been able to do it without a team effort. And again, to many of you that have contributed in the scientific community and that we've tried to build upon with our own efforts. So thank you very much for your attention.
Hi, elegant approach. I mean, finally figured out how to deliver STING, localized. So the question is, how low of a level of HER2 do these tumors express? So are they HER2 low tumors, or are they medium, or?
Yeah, the SNU-5 model is, I think, only about 20,000, 30,000 copies of HER2 per cell. So some of them, again, we did a whole range, but even down to very, very low copy numbers, we saw activity. We think it's one of the beauties of a STING agonist approach. In principle, with a cytotoxic, you're only going to kill a cell that you've delivered a cytotoxic agent to. With STING, what we're looking to do is start a response, right? If you can just spark a response, activate some of those immune cells, they can recognize, they can then take over. So we think that potentially this has applications where we didn't need bystander activity, because ultimately it's not our STING agonist that's killing the cells, it's that activation of the innate immune system that is giving us the anti-tumor effect.
Yeah, precisely the reason behind this is how low, like the previous talk we talked about, the HER2 lows, that's where the majority of the patients are, right? So if you can match to that and show the immunotherapy, the effect, right, that can really benefit this. So that's the question: how low can you go?
Yeah, and again, I think importantly, you saw the control ADCs. So if we didn't bind HER2, they were for the most part inactive, right? In that syngeneic model with the PD-1, you'll see there was some activity, but we do think target is important, but only low levels of target hopefully will be sufficient.
Okay, thank you.
I noticed that you had in your linker, you had an ester as their cleavable motif. Is that just an esteric hindrance is what controls the release? Is it meant to be just kind of a esterase kind of releasable mechanism?
Yeah, no, it's a great question. It is a novel linker with the ester. Importantly, and a lot of people think, esters, they're circulating esterases, how stable will that be? In the context of this, with the steric hindrance, et cetera, we see it's highly stable in circulation, but is effectively cleaved upon internalization. We do not know the precise enzymes that might be doing that, but that's something we'd like to understand better.
Empirically found?
Pardon?
Empirically found?
Yes. And as I touched on, it was only one slide, but we looked at many different linkers, and we found that one behaved very well for us.
Thank you for the talk. I was just a quick follow-up to the question. PKF profile you showed looked like there was quite a bit of loss of the payload, maybe 1 log in, I think it was in HP studies. And second question is if you have observed other formation when you do repeat dosing.
Yeah, sorry, this slide, you're talking about the data on the right. No, what we're measuring here is the antibody at the top and the conjugated payload on the bottom. You'll see those lines are parallel. So that difference that you see on the y-axis is simply reflecting the different molecular weights of those analytes. So we're not seeing free payload release in circulation.
Okay, so that's not like remaining payload on the antibodies total. Okay, and the other formation, have you looked into after repeat dosing whether you get antibodies against the payload or?
Against the payload or against the ADC? What I can say is, based on the preclinical studies, the ADA was no different than what we saw with cytotoxic ADCs, et cetera.
Okay, thank you.
Question. You'd mentioned that one of the driving factors for your drug is the interaction with your natural killer cells through the FC receptor. Have you noted any effects on those cells directly as they're taking up your STING payload? You'd mentioned that they don't affect the immune cells directly, but we study that?
So we do believe, and we have done studies where FcR gamma binding and internalization leads to activation and cytokine release from those immune cells.
So that's helping driving further killing by activating those STING cells?
That's right. In fact, in a very interesting and elegant experiment that Nanya Settenbud did, one of the questions might be, well, there's immune cells throughout the body. Why aren't we binding Fc gamma in the periphery? But it's that avidity enhancement that when you bind HER2, you're organizing the antibody on the tumor cell surface, and that organization and avidity enhancement is driving that Fc gamma into tumor-resident immune cells, whereas in an isolated immune cell, that FcR gamma interaction is not avidity enhanced. It's less efficacious, and therefore we can avoid that. And what was really interesting was showing the target dependence of that. If you just put our ADC on immune cells, we don't see much activation, even though there can be Fc gamma binding. But if you coat the plate with trastuzumab, then you organize by binding the with HER2. Then the ADC binds the HER2, it's organized, and now you see much more potent uptake and activation of those immune cells.
Because are you strengthening those immune synapses when you're doing this? Like when you're coating with the trastuzumab, you're recruiting all those HER2s and then adding your off-bind site to further drive that killing? Is that?
Yeah, I don't, as I pointed out, I'm a synthetic organic chemist. I'm not an immunologist. Nanya, if she were here, could answer that much better. But I think what we do see is that binding and avidity enhancement is a very important aspect of the behavior of the molecule.
Thank you.